So when I was asked to speak about precision medicine or stratified medicine, personalized medicine, genomic medicine, tailored medicine, or whatever, or however you want to call this kind of progress in the field of atopic dermatitis, I think I would like in the next minutes just to give you a few key bases which explain you what we are speaking about. First, I think that the problem that we had over the past, and this has been mentioned in some previous talks, is following. We always have considered our clinical phenotypes as one disease, and it has been shown already in all the different talks that we have to change our mind and the mind of the researchers, that in fact we are probably speaking about umbrella terms more than on distinct entities. So we are speaking about umbrella terms which are embracing a large group of different subtypes of disease, which may sometimes represent individual disease, but in many cases probably a spectrum of one single disease with different kinds of mechanisms. And the problem that we had when we are looking at the populations is that we are, as we are doing currently in treatment, using exactly the same approach. It's the kind of one size fits for all, because we are trying, or at least our colleagues in epidemiology, trying to assess information by questionnaires. I think questionnaires are very difficult to interpret. Then we are always trying to relate everything to severity. That's a kind of natural behavior that we have as physicians. We always try to find the best statistical power, which is always needed. And the inherent problem is that in fact we have absolutely no clue on how about stratification of the different groups. In many clinical trials, for example, there have been a lot of deception because we had the feeling this drug does not work. There is one good counterexample in oncology, which is trastuzumab. Trastuzumab, the story of the drug development of this particular thing was they included all the women with breast cancer and they applied this drug. And they just had 20% or so on clinical response. So the upper management of the company say, we stop this project, that's not a good product. And a very, I would say, good-minded people then said, just a moment, but we have 20% of people who are very well responding to that drug. Even if we have 80% who do not respond, it is worse to look at these 20%. And this is, in fact, exactly the kind of things that the people should have in their mind when they are going to interpret the data of clinical trials. I will come to that in a few minutes again. So, doesn't work. No. So that's the problem that we have in epidemiological studies and most of the clinical trials. We are thinking according to the model one size fits for all. The goal of precision medicine is to reverse this kind of thinking, because we would like, and this is the ideal situation, we would like to catch up the right patient, to treat them with the right drug, at the right doses, and most importantly, ideally at the right time point. That is, in fact, that's the whole philosophy of precision medicine. The problem is, this is very easy to express, but highly complex in terms of implementation, and this has been shown in the previous presentations. So what I would like to do now, take the example of the atopic dermatitis, which is my favorite disease, and to show you what are the approaches that we have, where are we, and where are we going. So first, just a few words about the heterogeneity of the clinical phenotype. Everything you have heard before about rhinitis and asthma, you can just one-to-one apply this to atopic dermatitis. It's a highly complex clinical phenotype with more mild forms, with more moderate forms, and certainly It's not the vast majority, but we have substantial amount of patients with really severe forms. And here, classically, we see the unmet need in terms of treatment. They all have in common one of a few kind of aspects: the dry skin, which is typical for these patients, which is due to genetically inherited barrier dysfunction. They all have in common chronic inflammatory reactions with pruritus, and in 80%, probably more, we have an increased IgE, or I would say it's probably more relevant to look at specific IgE, even if the IgE level itself is normal. This just shows you how diverse the clinical phenotype is. And we as dermatologists are very happy. You cannot, or you need some devices to look in the nose, and the asthma guys, it's even more difficult. We are really lucky. We can see on the skin, we see the inflammation, we see the secondary lesions from scratching, we see erythematous reaction, and so on. So it should be easy, and it's not. The second aspect in terms of heterogeneity is the onset of the disease and the fate of the disease. It's really surprising to see, because in our mind, in the mind of most of the people, atopic dermatitis is the archetype of disease of childhood. This is far from being the case. This shows you just that of course, we have a whole bunch of kids starting with the disease very early, but we have a number of other subgroups of patients starting later on, at the age of 20 or later, and recently also, we are just about to discover a new group of atopic dermatitis patients. They start very early, in elderly, and we have no clue what is happening there. They have a typical atopic dermatitis, and some of them are reporting that they had the same disease in childhood. Then they have a kind of remission. The disease was sleeping all the time over years, or 15 years, and then suddenly, for reasons that we don't understand, the disease starts again. Or they have this disease really from scratch, never had problems with the skin, at least to their knowledge. You never know exactly what happens before the age of 3 or 4. But this just shows you again how heterogeneous, just looking at the onset of the disease, this phenotype can be. So, if we consider the very early phase of the disease, the archetypal phenotype of the childhood onset, what can we do in terms of precision medicine? Because so far, in terms of prevention of the disease, most of the people, in fact, again, tried to have an approach according to the one-size-fits-all approach. There were never a real stratification in terms of approach for prevention. There have been some kind of approaches trying to select some patients, but nothing was really, really convincing. And it's not surprising, again, when you go to the interpretation of the results, that you are not really convinced about the overwhelming results of any kind of prevention in this kind of approach. It's not surprising because, in fact, we are always looking at the whole population, and this makes no sense, to my knowledge. In fact, when you look at the typical natural history of the disease, I think you can have a kind of concept. You can consider, in fact, the phase 0 of the disease. So this is an individual who had never had the disease, ideally, newborn baby, just after birth, between 0 and, let's say, 6 to 8 weeks of age. Nothing is seen, no disease, nothing. It's what I call the phase 0. And then the disease starts with the typical lesions on the cheek, on the scalp, and some other places. But at that time, you hardly will see any sign of sensitization. Prick tests are negative, total IgE is normal, almost no detectable specific IgE. And while the disease is going to be chronic over the time, months, years, they get sensitized and they have the so-called full-blown IgE positive atopic dermatitis. And of course, all these individuals, or not all, most or a substantial part of these individuals have a high risk to go into the classical atopic march with atopic or with allergic rhinitis and asthma. And we now know that we have to consider a genetic background, the predisposition on one hand, but we should not forget the environmental part and of course the direct impact of the environment to the genetics by the pathway of epigenetic regulation. But the key point is we have to consider always a kind of phase zero, which is the phase, in fact, which is the ideal phase to provide some kind of prevention. I will just give you an example. 3 kids that I have seen in my clinics, they were all starting like this, very nice baby, newborn, nothing happens, the skin is beautiful. After a few weeks, all 3 came with the same clinical phenotype. They all had a typical form of, quite a moderate form of atopic dermatitis. They had no IgE, by the way. The one Petra, she showed really a complete remission, no IgE. Everything is perfect, life is magnificent. The 2 others, by the time they kept the disease, they started to be sensitized and had a classical atopic dermatitis with increased IgE. But Amelie lost the disease by the age of 8 years, and she still is suffering from allergic rhinitis because he has this specific IgE to grass pollens. The worst scenario was Jonathan. He kept the disease despite all the kind of therapeutical approaches we had. He kept the disease until now. He has a severe form of AD, he has asthma, he has rhinitis, he has a typical atopic march, and he regularly suffers from various complications that we call eczema hepaticum, which are due to the widespread form of herpes simplex. Now, could you imagine, and this is, the dream of all dermatologists and pediatricians, could you imagine that here at phase 0, you would catch up these kids and then to imagine something that would be targeted to these kids in order to avoid the appearance of the disease itself, with the idea that you can stop the whole story behind this. And this has been done in Japan and in the UK and in the States. I just show you the slide of this Japanese story, where they have recruited 180 newborns with, according to the family history, the highest risk to develop atopic dermatitis. They divided this cohort in 2 groups. One group in which the parents have not been educated to do anything in terms of skincare, and the other group where the parents have been asked to apply an emollient of their choice twice a day for at least 6 months. And you see here the result. This is the Kaplan-Meier curve showing you the probability without atopic dermatitis. And you see here this delta, which is statistically significant. So this shows you that in this seemingly selected groups, just based on the history of the family, but there is some kind of stratification here, you have a significant difference by this very simple measure, which is just using skincare for 6 months in a row. And then you have a difference in the appearance of the disease depending on this just very simple way. The problem we have here, I think we could be able to improve these results if we would have, for these newborns, something in our hands, either measuring the transepidermal water loss, doing filaggrin mutation and analysis, doing Raman spectroscopy or something else, which allow us to be more precise in the selection of those kids who really would have benefit of this kind of approach. So this is prevention. Now we'll give you an example in terms of therapy. Atopic dermatitis is like rhinitis and asthma. In some cases we are treating with steroids, in other cases we are treating with cortisone, and in other cases we are treating with something which is called glucocortisone. Very differentiated approach. Luckily we also have the topical calcineurin inhibitors as steroid-free kind of anti-inflammatory drugs. The problem is we are always treating the patients according to the same approach, more related to the severity than to something else. And the typical way to treat inflammation is using topical steroids and or TCIs. So the other point is that according to the classical understanding of the disease, we were for years always looking at the T2 cytokines because we were always believing that atopic dermatitis is a typical Th2 driven disease, which is certainly at least partly the case. And if this is true, you would expect that the new generation of treatment, which will be available in the States by the March 29th, by a decision of the FDA in next year, and probably in 2018 in Europe, once the approved has been given for that drug, is the use of anti-IL-4 and anti-IL-13 approach, blocking that seemingly, I would say, key cytokine in the disease atopic dermatitis. Now, if you look very closely at the response of the dose response in terms of scoring of these individuals, in that particular study, for example, which was published last year, you see here that as you're using your old clinical scoring in asthma and in rhinitis, we are using the so-called EASY scoring or SCORAD or IGA, depending on the kind of trials and the regulatory landscape. This is 50% improvement overall of the disease. And you see here, using dupilumab, that new kind, the first-in-class molecule targeting IL-4 receptor and IL-13 receptor, you have 85%, 90% of the patients who have reached 50% improvement. I mean, this is really a progress for us, you know, using something which is not a steroid and reaching this kind of progress for the patients who are really suffering, heavily suffering from that itching. They have a high impact on the quality of life, really a burden for these patients. This is really progress. But if you would like to have something which is working in all the patients in the way that they are, as we call, clear or almost clear, so we are speaking about true remission, and this would be something that we would expect in what we call the EASI-90 response. That is, the patients have more than 90% reduction of the clinical scoring. Then the situation looks very different. You really have here something which is peaking at 40%, and we are far from what we would like to see if we would treat all the patients according to the one-size-fits-all scheme, that all the patients have that kind of nice response. There is another disease in dermatology, which is psoriasis, where with the new kind of compounds targeting IL-17, typical example is Cosentyx, you reach here the so-called PASI 90. And with the newest molecule, we have PASI 100. This means you are treating these patients with these new drugs, and they are completely cleared. There is nothing to see for a longer period of time. We are far from that. So the question is, we believe here that we are targeting a key cytokine, but still, we do not have the full-blown response that we would expect. I mean, this is really progress for us and for our patients, but it's not the magic bullet, to be clear. Why it is this? You may imagine different kind of explanations. The role of the barrier function, which is not considered here because you are targeting a pro-inflammatory cytokine. The need for combination, why not? Or, and I think this is most important, we forget here the disease heterogeneity. Again, the same problem. Do we have answers to this question? I think yes. First answer is coming from that paper from Emma Gutmann and co-workers. They have shown that in fact, in terms of dominance of cytokines and related to the age, comparing the kid situation and the adults, there is a difference. The assumed Th2 dominance is more, let's say, present in childhood than in adulthood. In adults, we have a more Th22 dominance, while in kids we have the expected Th2 dominance. And this shows you how, again, at the level of immunological response, the complexity of this phenotype. And since that studies have been done in adults primarily, this is according to the regulatory rules, not yet in children, I think it's not surprising that you have this rather low response. My expectation is once we have more insight into the pediatric population treating these kids with that particular product, the response would be much higher. Another point in terms of heterogeneity of the phenotype from a mechanistic point of view is that, and this is quite recent also, we have to realize that although we are speaking about atopic dermatitis, the phenotype, the clinical phenotype, and obviously also the immunological response behind this seems to be different looking at different ethnic populations. When I'm going to China regularly and I look at patients suffering from atopic dermatitis, I always got the impression, hey, these guys look a little bit different from our patients, our Caucasian patients. There is something different. They have more lichenification, the overall involvement is different. It's not the same disease. It's called atopic dermatitis, but phenotypically, it's different. And, these guys, again, the group of Gutmann have shown that in that particular population, in Asian population, it seems to be there that beside the Th2 component, there is also a Th17 component, something which is in the Caucasian population more related to psoriasis, as I mentioned before. So this is another kind of explanation why in clinical trials targeting one particular cytokine, we may not have this kind of 100% response. So this is exemplified here where in this recent paper from Stefan Weidinger and Natalia Novak from my group, they have again shown that in fact beside the Th2 response, there is much more behind this from an immunological point of view. Th22, maybe Th9, maybe Th99, who knows, something which may be the complementary information that we need. For me, very much involved in drug development programs, looking at what is doing the pharmaceutical company, I'm very pleased to see that in fact, against what happened 10 years ago now, we have a whole bunch of new targets and different approaches. The problem for me still is that all these companies are always looking to this, according to the same one-size-fits-all. Of course, they have their business model. They want to have most the possible patients to treat with that. But I'm not sure that this is really the right response. So they probably, in the context of clinical trials, they have to look at the subgroups. They have to stratify their individuals. And how to do this job the best? The best would be to have the stratification based on biomarkers. And this has been nicely explained. Currently, what we try to do basically is we have this very heterogeneous population with seemingly the same disease, and we try to use basic on biomarkers to create more homogeneous subgroups. And ideally, at the end of the day, we have a kind of umbrella term designing the disease, but this will be, in fact, we will add a kind of molecular taxonomy to that umbrella term, which gives us the information about different kind of aspects. And there is not one kind of biomarker. There are several kinds of biomarkers. Biomarkers for the diagnostic, biomarkers for the prognostic, biomarker predictive for the biological response, biomarkers predictive for the fate of the disease. And when you look currently at the literature trying to see what is the status, what is the state of the art in terms of biomarkers trying to answer these different kind of questions, you will be very, very disappointed because most of the biomarkers currently available, they are not validated. None of them are validated, by the way. They are just candidate biomarkers. They are almost all looking at severity. For me as dermatologist, this makes no sense. I see the patient, I see the skin, I can tell you how severe the disease is. I don't need a biomarker to express the severity. What is more interesting for me is to have a biomarker to screen the right individuals in terms of prevention. what makes more sense is to have some biomarkers looking at the predictive response to a treatment. Nothing available here, nothing. And also in terms of prognosis, fate, comorbidities. We have some few biomarkers, but again, the problem that we have is that we have no validation for that. And what we are currently doing in the CK Care Consortium, to which Chesme is also contributing, we are currently collecting material with the patient informations in a huge registry of the patients. We plan to collect several thousands of individuals, and we are collecting the biomaterial of these individuals in a biobank and linking these two in order to be really able in the future to have the answers, to have the real biomarker for these different kind of categories. And then, and this is another exercise that we have to perform, and Joanna has mentioned this already, we are here, we are at the identification stage, we are at the biomarker discovery stage. None of them has been validated so far. And none of them has been qualified, for example, as a surrogate marker. What I think will be in the future, because this has been mentioned before already, we are treating our patients according to guidelines. Please consider one important aspect in terms of guidelines. For years, we treated our patients by what we called empirical medicine. In the last 20 years, due to the economic pressure, we introduced the so-called evidence-based medicine. And according to that, we also were more and more looking at one-size-fits-for-all strategies. In the future, when precision medicine will be established, we will have a widening of this bottleneck, again, with different kind of guidelines, which will be, personalized and adapted to the different groups of the different phenotypes. Because my dream is that the phenotype atopic dermatitis in the future, according to what we will know about the pathophysiological mechanisms and all the biomarkers and endophenotypic information provided from them, we will be able to dissect that complex phenotype into different subgroups, and these different subgroups will then be, in fact, be designed, or for these subgroups, different kind of strategies in terms of prevention and in terms of treatment will be designed, including something that we currently are designing, orphan diseases like the Netherton syndrome, which for me is nothing else than the maximal variant of an atopic dermatitis due to a null mutation in the SPINK5 gene. So this is, just a glimpse of what is coming to us as dermatologists, and I think this kind of approach could be applied for asthma and rhinitis as well. I hope I was able to show you a little bit what is going on in terms of precision medicine for that kind of paradigmatic disease, which is atopic dermatitis. And as you see, the progress is very slow. We are at the very beginning of a fantastic story. But we have to engage a lot of efforts and investment in the development, in the discovery of biomarkers and huge studies in order to be able to stratify the patient. Thank you very much for your attention.